Hi everybody, welcome to our module on shapes and stains. In general, there are basically three methods we use to identify bacteria that cause infections. First, we look at their shape, then we look at the color they take on after certain stains, and then finally there are lots of special tests like DNA tests and antibody tests. But just looking at the shape of the bacteria and the color it takes on after staining are very, very important. These are time-honored old methods of identifying bacteria, and they're still useful in the modern era. There are some common shapes of the bacteria that cause human infections. The first common shape is called a concise or a sphere, and bacteria that have this shape are called cocci, like what I've drawn over here on the screen. Another common shape are rods, which are also called bacilli, and rods look like what I've drawn over here. And then there are some bacteria that are somewhere in between. They're sort of like a sphere, but also sort of like a rod. We call those coccobacilli or coccobacillus shaped bacteria. And then finally there are lots of other shapes, but these are certainly the ones that are very common right here on the screen. When you have more than one cocci on a slide, they tend to form certain characteristic patterns. The first pattern is for them to be grouped in pairs. This is called diplococci. The next pattern is to form chains, and Streptococcus tends to form chains. And the reason this happens is because each new bacteria forms away from the older bacteria so that you get them forming away from each other and forming these long curved chains. And then the final pattern is to be in bunches or clusters, and that's what Staphylococcus does. It forms clusters like this. Sometimes they're described as clusters of grapes. And on this slide I've just shown you some microscope pictures of what they might look like. So on the left side of the screen we have some Streptococci in chains, and you can clearly see that there are little circles forming long chains here, and that's what Streptococci look like. On the right side we've got little circles, but they're forming in bunches or clusters, and that's what Staphylococci tend to do. Here are some rules of thumb about cocci that may be helpful to you. Most cocci are gram-positive, so we're going to talk about the gram stain in a few slides, but for our purposes here, just know that bacteria can be divided into gram-positive and gram-negative bacteria. And most cocci are gram-positive, so most cocci are either strep or staph, and these are gram-positive bugs. Very few gram-negative bugs are cocci. There are only a couple that are. Neisseria, either meningitis or gonorrhea, are gram-negative cocci. And then another bacteria called Moraxella is also gram-negative. But in general, when you hear about cocci, usually it's going to be a gram-positive bacteria. Now let's talk about rods, which remember are also called bacilli. So most rods and most coccobacilli are gram-negative, and gram-negative rods are very severe infections that tend to make people very, very sick. There are just a few gram-positive rods. So Corynebacterium, which causes diphtheria, is a gram-positive rod. So are Clostridia and Listeria and the Bacillus species, which cause anthrax, and B. cereus, which is a foodborne infection. But other than these couple exceptions right here, most of the times when you hear about a rod, you're talking about a gram-negative bacteria. Let's talk about some of the other shape that bacteria sometimes take on. So some bacteria have long branching and filamentous structures. There's a picture of one over here on the screen. And this looks like fungi, and the two bacteria that do this are Actinomyces and Nocardia. And in fact, when these were first discovered, people thought they were fungi. It was only later they determined that they're bacteria. Spirochetes are bacteria that have a long twisting shape. There's a picture of what one might look like here on the screen. And there are three spirochetes you need to know about. We have a separate module on those. Treponema, which causes syphilis, Borrelia, which causes Lyme disease, and Leptospira, which causes leptospirosis. And then finally, Vibrio has a very unusual sort of curved shape. I'm drawing a circle around it on the screen right here. It's often described as a curved bacteria, and Vibrio is what causes cholera. There are two bacteria that are described as pleomorphic. This means they can take on many shapes, and these are Rickettsia and Chlamydia. And it's nice to remember these together because they are the two intracellular pathogens. These are obligate intracellular bacteria, and in addition, they are also pleomorphic. So for the remainder of this module, what I'm going to do is talk about common bacterial stains, and far and away the most important is the gram stain. We'll talk about that in detail. And then I'll also mention the Giemsa stain, the Zeal-Nielsen stain, the silver stain, and finally the India ink stain, which is used not for bacteria, but for Cryptococcus, which is a fungi. So before I mention specific stains, you should know that there are a lot of simple stains that we use just to see bacteria under the microscope. So over 100 years ago, when people figured out that bacteria cause disease, they had trouble seeing them under the microscope. So these stains were developed which stain basically all bugs the same. And these are things like methylene blue, safranin, and crystal violet. You add them to a fixed preparation of bacteria and then wash it away, and the stain remains behind. And these are non-specific, but they just allow the bacteria and the bacterial shapes to be easier to see. So the most important stain from a medical point of view is the gram stain. It identifies bacteria as either so-called gram-positive or gram-negative. So if you look at this picture on the screen, you'll see some bacteria, like the ones I'm drawing a line around now, which form purple circles, and those are gram-positive bugs. You can remember that gram-positive has a P for positive, and P is for purple. They become purple on the slide. Some of the bacteria on this slide are also red or pink, like these ones I'm circling over here, and those are gram-negative bacteria. And we're going to talk about this in a minute, but the bacteria stain differently with the gram stain based on characteristics of their cell wall. So the way you perform a gram stain is you fix some bacteria on a slide, and then you add crystal violet, and then you add iodine. And after you add the iodine, all the bacteria on the slide are going to be purple. But then you add a decolorizer, and it washes away the purple color from everything except the gram-positive bacteria. They will retain that purple color, whereas the gram-negative ones will not retain it, and they will turn colorless. After you do this, you add safranin, which is called a counterstain. It won't do anything to the gram-positive bugs. They will stay purple, but the colorless gram-negative ones will then become pink or red from the safranin. And this is the way you do a gram stain, and at the end of the stain, the bugs will either be purple if they're gram-positive, or pink or red if they're gram-negative. So just to summarize some of these key points, remember the purple bacteria are gram-positive. That's the P's for purple and positive, and that's because they retain the crystal violet in their cell walls. The red or pink bacteria are gram-negative. They do not retain crystal violet in their cell walls, but they do take up the safranin counterstain. And it's very high yield to know that the thick cell wall of peptidoglycan in gram-positive bacteria is what makes them hold on to that crystal violet and turn purple. And I talk about this in more detail in the module on bacteria. Now the gram stain is not perfect. There are some bugs that do not gram stain well, and you should know what they are. One of the first ones is treponema, which is the causative agent of syphilis. It's a spirochete, and it's too thin to see, so it doesn't gram stain very well. Mycobacteria, especially tuberculosis, do not gram stain well, and that's because they have mycolic acids in their cell walls, and we use special stains called acid-fast stains for mycobacteria. Mycoplasma has no cell wall, so it doesn't gram stain well. And then finally, intracellular bacteria don't gram stain very well because they grow inside of other cells, and it's hard to get them to even grow on a plate, let alone to get the stain to be taken up by them. So rickettsia and chlamydia are obligate intracellular bacteria. They don't gram stain well. In addition, chlamydia has no muramic acid in the cell wall. Muramic acid is one of the two components of peptidoglycan. And then Legionella does not gram stain well either. It's mostly an intracellular bacteria. Next stain is called the Giemsa stain, and this is a mixture of methylene blue, eosin, and azure B. And its discoverer was a guy named Gustav Giemsa, and in the late 1800s and early 1900s he was looking for a way to visualize Plasmodium, which is the protozoa that causes malaria. And you may know that Plasmodium is intracellular. It lives inside hepatocytes and red blood cells, and if you can remember that, it's an easy way to remember that the Giemsa stain is most useful for intracellular findings. In addition to infectious disease reasons, we often use it as a stain for other things like blood smears and bone marrow because it'll get inside the cells and stain the nucleic acids. From an infectious disease standpoint, though, you can just remember that the Giemsa stain is used for protozoa like Plasmodium and trypanosomes like the bacteria that cause Chagas disease and sleeping sickness. And it's used for some intracellular bugs like chlamydia and rickettsia and borrelia, which sometimes is an intracellular bug. And you can just remember that it goes inside cells. It's a useful stain to get inside of cells. So Plasmodium lives inside cells. So do chlamydia and rickettsia. And that's the way I always keep straight the utility of the Giemsa stain. Ziehl-Neelsen is the acid-fast stain. It contains a substance called carbofuscin, and this substance is used to help you visualize Mycobacterium, especially tuberculosis. It also is used for nocardia, and what happens with this stain is that acid-fast bugs resist decolorization with acid solvents. So this is a picture of some acid-fast bacteria here, and they are these purple bacteria. And initially when you stain a slide with this stain, everything will be purple, but then when you decolorize it with an acid solvent, only the acid-fast bacteria will retain that purple color. And the main use of this is in detecting tuberculosis infections. Silver stains are very rarely used stains because they only have a couple of special circumstances where they're helpful. The first one is in pneumocystis pneumonia, which patients get when they have severe or end-stage HIV and AIDS. It's a fungal infection that causes diffuse interstitial pneumonia, and it's very hard to see this fungi unless you use a special silver stain. Legionella can also be stained with silver stains. It causes interstitial pneumonia, it contaminates water, it classically causes outbreaks in nursing homes. It's very hard to visualize Legionella, and so these special silver stains are used. And then the third example is H. pylori, which you may recall is the causative agent of gastric ulcers. So for a long time, people didn't think that gastric ulcers were caused by bacteria because nobody ever saw any bacteria under the microscope. And that's because H. pylori is hard to identify, but silver stains were one of the ways that were discovered that allowed people to visualize the bug. The last stain I'll mention is called India ink, and this is a stain that's not used for bacteria, it's used for fungi and yeast. India ink is a negative stain. What it does is it makes the background very dark, not the organism, and therefore the unstained organisms stand out in contrast. Its primary use is to identify Cryptococcus neoformans. Cryptococcus neoformans is a fungi that exists as a yeast, and it has a large polysaccharide capsule. So when you stain the background dark with India ink, like I've shown in this slide right here, the yeast will stand out, and you can see that they are these small circles with little halos around them. And the reason they have the halos is because of their large polysaccharide capsule. The last topic I'll discuss in this module is that of pigments. You should know that there are some bacteria that produce pigments as they grow, and this gives them a special and unique color. The first one to know about is Staph aureus, and the word aureus actually means golden, and that's an easy way to remember that Staph aureus produces a golden yellow color as it grows. Pseudomonas aeruginosa produces a pigment called pyocyanin, and this gives it a blue-green color. Serratia produces a red pigment when it grows. And then finally, actinomyces is a filamentous bacteria that cements together as it grows. So it's a long, branching bacteria. It sort of looks like a fungi, and in fact, when it was first discovered, people thought it was a fungi, but it turns out it's a bacteria. And the colonies of cemented together actinomyces have a yellow-orange appearance, and they're called sulfur granules. And actinomyces is a normal oral flora, but it sometimes forms abscess infections in the jaw, and you can sometimes see this yellow sulfur granule material draining from the abscess. Here are some pictures of what those look like. So here's Pseudomonas growing, and you can see it's got this bluish-green sort of pigment on the slide. This is some Serratia growing on a plate here, and it's got all this red pigment in the middle of it. And then finally down at the bottom, here's some Staph aureus with that golden sort of yellow color as it grows. And that concludes our module on shapes and stains.